Some of you have pretended as if I didn't give you an assignment. I haven't received my assignment. You haven't finished it. You brought it yesterday. Make it into a PDF and send it to me into my email, or send it on to Dillis, and then I'll assess it. And we'll give extra pizza to the ones who win at tonight's dinner. All right, great. So we want to make a quick start to this. So far, just by way of revision, we've tried to take you through light. We've done the basics and the fundamentals of light. We've done some basics and fundamentals of the eye. And then we took you through to some data, evidence synthesis, how we actually arrive at the things that we use our clinics. And yesterday I heard everybody managed to do a Gable patch. Did everybody manage it? Clap for yourselves, clap for yourselves. Yes, if there's anything you can take from it, I have never designed a Gable patch before, even though I asked them to teach you how to do it, but I haven't done it. So this is one thing that you, hello? Maybe 26, if some of you, or call you as facilitators, and so you how to do Gable patches. Try 1, 2. Okay, maybe we'll go with this one. Yeah, so today we dive into electrophysiology. You should all have received 2 reference documents into your WhatsApp pages now. You can download them. So they are for reference. Somewhere in the slide, I would mention them. It's quite a lot. So there are 2 of them. One is more like a textbook that gives you all the details that you would need to do electrophysiology. It can be very technical. And then the other one is a write-up that we wrote for practitioners. So a colleague of mine in the UK and we wrote that version for practitioners. So I'll say you can start with that one. So treat that one as milk, and then when you are fine on the milk, you can go to the bones and try that one. But let's get into today's business. Oh yeah, those are my bosses. Yeah. Okay, so for those of you who are clinical, I'm sure you've come across this. This is an OCT. So this is a scan of the, we say the back of the eye. So a scan of the retina. So you have a 29-year-old male. The prescription is minus 0.75. So if you are thinking high myopia, forget it. It's just minus 0.75. And the patient comes with this kind of cupping, okay? You do your OCTs, and that's what is giving you here. So that's your markers. And it says here that the right eye CD ratio is 0.6 or 0.55, and just for comparison, the left eye is it's just also like 0.6. The person is 29-year-old, like this, and it says within normal limits. I think this was minus. Do I remember what it was? I think it's minus 2 something. Okay, that's what your mean deviation is looking like. You've got these uncertainties here, one around the, how do you call it? The blind spot. What are you going to do about it? Do you refer? Do you monitor? What do you think you are going to do about it? You are just going to monitor, yeah. So, but it would have been cool if you could tell definitely, okay, what was the issue. So the challenge we have with these tests is that OCT is objective. I mean, you take a picture of the structure, but it doesn't tell you the function. Of course, you can make inferences about the function. I mean, if you see a healthy person, a healthy looking person, you expect that the person will be healthy. But if the person has HIV, the person will look healthy, but the person is not all there, okay? Then we have a functional test, which is the VFT. You check the function, but it doesn't tell you, it's not objective. It depends on the person pressing the button, right? If the person is not telling you the truth, what do you do about it? So it would be nice to have a test which is both objective and both functional. And that is where visual electrophysiology comes in. So it is an objective functional test of the eye. So today, we jump into the basics. All right. So the wonderful thing about the human body is that it's an electrical generating power plant. Okay. So we are used to doing electrocardiograms. We know that one. If you've watched enough movies, you've seen somebody rush to the theater and then they put some things on their chest and they do they beep, 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 beep. Yeah. It's basically just giving us a signal of how the heart is running around. And then we have the ones for the head as well. Those ones are more common. But when people ask me, what do you do? And I say, I measure electrical signals from the eyes to the brain. They're like, is this guy from space or something? Yeah. So the same thing that we can do, we can measure these biopotentials. We can measure these biopotentials in the body. And we can measure it from the eye to the brain and at different points along that path. So how does it work? Remember, light enters the eye. Oops, sorry. So light enters the eye and then does whatever it needs to do, gets to the retina. At that point, it is they are photons. The photons then go through the retina and they are transformed into a chemical signal first and then an electrical signal through the process known as phototransduction. All right. So phototransduction occurs at the photoreceptor layer. That's where you have the light energy being transformed into a chemical signal. The neurotransmitters are sent through to the bipolar cells. The bipolar cells pass them on to the ganglion cells. The ganglion cells then transmit it to the brain as electrical potentials. And we are able to measure the process in the retina by sticking electrodes. Okay. Now, if you want to measure these signals, ideally, what we should do is take some 2 pins and stick them into the retina. But nobody will let you do that. So what we do instead is that because you have a fluid surrounding our tissues and water is a wonderful conductor of electricity, we can tap into that electrical flow from somewhere a little bit away from the actual generating source. It comes with some problems, but there are workarounds it, okay? Because now if you are tapping it slightly away from the actual source, then there are other parts of the body also generating signals and you have to kind of take it out. So those workarounds are what make electrophysiology unique and also very expensive and also very technical, because you need to be sure that you are measuring the actual signal that you want in spite of all the things that are going on there. So when you get down into the lab, you would realize that we will talk about things like electrode placement. Where's my staff? Okay, I think they are down. Oh, there she is. Okay, so you'll be meeting there. So Dillis and Ima take you through the electrodes, like those 2 pins that we should have stuck into the eye, where we would put them and what they actually do there. Wonderful. All right, in electrophysiology, what we are also measuring, so you get a signal. Basically, you just get a signal that looks like this most of the time. All right, and we are interested in the amplitudes or the height or the size of the signal and the speed at which the signal is moving. There are 2 technical words you need to keep in mind. One is the amplitude, and the second one is the implicit time. The implicit time just tells us how quickly that signal is moving. Oh, yeah, okay, great. The Electrophysiological techniques, sometimes we abbreviate it as EDTs, come in these three general forms, all right? So the first one, the electroretinogram, measures the signals from the retina. And here we are interested in the rods, the cones, the bipolar cells, the retinal ganglion cells. Can I ask a favor? Can you turn, is that screen on? Yeah, can you just turn that one on? Turn it towards me, please. Yeah. And then the electrooculogram gives us, wonderful, okay. The electrooculogram gives us signals from the retinal pigment epithelium and the outer retina. And then the visual evoked potentials give us signals from the cortex, right? We are mostly measuring from the cortex, but it relies on the macula being very healthy, okay? That the macula can pick up the signals. A thought just occurred to me. In ophthalmology and optometry speak, we often talk about the inner retina and the outer retina, and it can get very confusing. Which one is the inner retina and which one is the outer retina? Okay. Anything that is towards the inside of the eyeball is inner, and anything towards the outside of the eyeball, that is like the external, the sclera, the orbit, and all those things are outer. Now remember that the retina is turned the other way around. So the photoreceptors are more towards the sclera, and so they are in the outer retina, while the ganglion cells, which are inside towards the vitreous, form the inner retina. So this, you need to become very familiar with this terminology because we'll be using it a lot. Okay? Yeah. Okay, so let's start with the electroretinogram. So in the electroretinogram, we are looking at transmission of impulses from the photoreceptors, the bipolars, the retinal ganglion cells. That's what we are interested in. What happens is that you show a stimulus, or just as the stimulus is just a light source. You show a light source to the eye. You know the intensity of the light. You know the rate at which that light is blinking. You can tell what is going on inside the eye once you remove everything else. Okay, so if you imagine yourself in a marketplace, let's take Kotokraba, Kejetia, or wherever, all right, and you are trying to listen to the sound of your ticking clock. Okay, all right. In the midst of all that noise, how are you going to do it? Yeah, let's think it through. You want to hear the sound. Good, yeah, that is one thing. Would be to bring it closer to your ear. Yeah, all right. Now, what are you listening for? There is something intrinsic about the ticking clock that is not in the environment. What is that? What would you hear? You would hear the ke. In fact, you anticipate it. You expect to hear that ke sound. All right. Okay. So what your brain is doing is that your brain has shut down all the other noises because they are random. They are random in the system, and you are listening to just that sound. All right. So in electrophysiology, we program the computer to listen out for the signal that matches the stimulus we are generating. You get it? So even though there will be all other things going on, and when you get in, you see, for example, you blink, you clench your teeth, your body would jitter a little bit and all those signals, because the computer has been instructed to only listen out for that regular pulse. It would sort of average out, we call it averaging, so it will average out all those random noises and only try to pick out the signal that is coming out from there. Of course, there's the possibility that somebody will have a very, very big clock at edum while you are at KJTR, which will also exactly be at the same time as your clock, right? So there can be other confounders, but generally it works. Okay, so that's how that works. Why is one of my diagrams missing? Great. The other thing too about electrophysiology is that we can vary parameters of the stimulus and we get different signals. So if the light flash, we call it the duration, so if the duration of the flash is very short, so it goes, right, you would get relatively short, you get a short signal. You get just this part, okay? All right. Now we can also make the light stay on a little bit longer. And when it stays on a little bit longer, the eye does something called the on and off switching. So I think somebody talked about on and off. Yes, good, inside the eye. Okay, so the system comes on and then goes off, and the signal, the signal that is generated by the off pathway will be seen, and you can measure that as well. When the flash is very short, there isn't enough time for the off signal to be properly separated from the on signal, so they merge and you get what we call the brief flash ERG. But you can also separate this, and this is important in certain diseases. There are certain diseases that can affect only the on pathway, but not the off pathway of the retina. All right, and so if you suspect that, then you use a different kind of stimulus to make that separation. But for the most part, we just do what is called the brief flash ERG. There is a body or an organization known as the International Society for Clinical Electrophysiology of Vision, ISCEV. And ISCEV has what we call standards, like protocols, that should be used generally. Of course, you can do other different kinds of stuff, which we call the extended protocols. So I am part of that organization. Professor Sven Heinrich is also part of that organization. In fact, he has been very influential in writing quite a number of the standards. The lab that I went to work in is famous for writing a number of the standards. So when he talks, please listen. He knows what he's talking about. Yeah. And they have set out a number of tests that need to be done. So they are called the ISCEV standards. Later on, you see a video, and my colleague would reference that. So the second pamphlet, the big textbook-like one, is the ISCEV standard. Okay, and it quickly goes through the various things. So there's a standard for dark adapted, and I need to mention this as well. We know night blindness, right? So people with night blindness, they can't see at night. They are fine in the day. Then we have albinos who are fine at night and have problems in the day. All right. So if you are testing certain conditions, you have to test it in these dark adapted conditions and light adapted conditions. You'll be hearing the word photopic and scotopic. So photopic has to do with photosynthesis, light. Scotopic, I don't know. I thought of scopatomana, but I don't think it's the right thing. All right, okay. So they have recommended a certain number of tests for the dark. So if the eye functions very, very well in the dark, then there is a standard amount of light that when you flash it, the retina should respond and you get a signal. And they call it the dark adapted 0.01 ERG. Then they increase that amount of light a little bit, and then they have a third one where they blast the whole eye with light while you are still in the dark. Then they have oscillatory potentials, I'll come to that, and then the light adapted ERG. And then they can also make the light flash very quickly and see how quickly your eyes respond. Think of strobe lights, you know, the disco lights, how well your eyes respond to things like that, and then they measure it. Something I didn't mention earlier, but there's no problem going back now. The ERG wave, and I'm spending quite some time on this slide because when you get this slide, the rest are easy to follow. So the ERG wave has this negative dip, okay, or trough, right? This bit here, which is called the A wave, please, scientists, we are boring. Don't ask me who decided on calling it A, but it's called the A wave. This A wave signals photoreceptor activity. All right, so a nice deep wave here means your photoreceptors are fine. This hill here is called the B wave, and this one corresponds to the bipolar cells. This one is not called the C wave. There is a C wave, but it doesn't show up in this one. This signal down here is called the PHNR. This is actually relatively new. It was discovered around 2000, the year 2000. ERGs have been around since the 50s or 40s. I would have to check my history. But this bit was much more recently discovered. I mean, it had always been there, but they didn't know exactly what it was signaling. So it was some work that was done that helped us figure out what this side represents. So it's known as the photopic negative response. And the photopic negative response corresponds to the retinal ganglion cell function. Okay. Now, I mentioned something called the oscillatory potential. So this is a drawn out ERG, right? This is like your perfect of perfects, okay? The ones you will see down there will not look like this. They will have all sorts of wiggly things on them. You will see the general shape, but it will not be this smooth, all right? Let me finish up. So if you have a long duration stimulus, then it flips the other way. You are not going to get a new photoreceptor activity. It's just the bipolar cells which are responding. Remember, the on and off system starts in the bipolar cells, okay? So now the off bipolar cells now kick in. So it's known as the D wave. And then when the signal goes down, you now have the off retinal ganglion cells kicking in and giving out the photopic negative response, but this is in the off pathway. So just to go over it again, we have the A wave, which is photoreceptor activity, the B wave, which is bipolar activity, and then the photopic negative response, which is retinal ganglion cell activity. That is when you have a brief flash. If you use a long duration flash, and then you are able to expose the off pathway, then it continues to the D wave, which corresponds to the off bipolar cell system, and then the PHNR, or the photopic negative response, the retinal ganglion cell off system. All right. The oscillatory potentials, if you look in this diagram here, and this is where, if you have your textbook, if you're able to access your text, there's a bigger diagram in there. But otherwise, just you can follow here as well. You can see that there are some sort of small bumps on the rising edge of the B wave. Okay? All right. They are thought to represent inner retina activity, most likely the horizontal cells. All right? Okay. And so when you have healthy ones, then you know that the horizontal cells are functioning very well. Okay. So, as I indicated, the ERG, which has to do with the retinal function, comes in 3 flavors. So we have the full field ERG, where essentially you have the person, you are trying to get the overall function of the entire retina. Okay, so you might come across the word Gansfeld. or Gangsfeld bowl. It is just German for the whole field. All right. But English people like borrowing other people's language, so you'll find it in textbooks as the Gangsfeld ERG, but it just means full field ERG. So you can, of course, have a focal field ERG. All right. Then there's the pattern ERG. Now, the pattern ERG, instead of using a flashing light, what you have instead is a checkerboard, all right, that alternates while the person is viewing it. And in this checkerboard, it focuses, it's mostly taking signals from the macular region. So the pattern ERG is used to check macular function. Again, remember that we are talking here about the retina. So we are talking about the retina, but we are looking at just the macular function. By the nature of the stimulus, which is a check size, the part of the retina that mostly, that mostly responds to check sizes is the ganglion cells. Okay, so the ganglion cells respond very, very well to check sizes and borders and stuff like that. Okay, so the pattern ERG is mostly used to assess retinal ganglion cell function in the macular area. And so again, you get a wave like that. There's the N35. It doesn't mean much. Don't worry about it for now. What we are interested in is the P50 and the N95. You are going to be coming across these numbers and letters and all of that. Again, scientists are boring. The P simply means positive, and the 50 means after doing this experiment in several people for a long time, the average time at which you see this positive peak is 50 milliseconds. Anybody wants to tell us what the N95 means? The negative trough at 95 seconds. That is all that it means. Okay? All right. So this P50, so the whole pattern ERG gives us an indication of the retinal ganglion cells. But you need the photoreceptors to drive the signal. If you don't have photoreceptors, forget, it's not going to go to bipolar, it's not going to go to ganglion cells. So a bit of the photoreceptor response is captured in this P50. And then the N95 represents most of the retinal ganglion cell. Can you think of any disease that is retinal ganglion cell? A very common disease, that glaucoma. So glaucoma is your number one thing when you are looking, or any of the retinal ganglion cell diseases. So the pattern ERG is used mostly for glaucoma test. I didn't give you any disease for the first one, but if you are thinking of things like diabetes, which disrupts activity inside the retina, that's when you want to use the ERG, if you so desire. The last one is the multifocal ERG. Okay. Now, this also a newer technique, and basically, it was produced because there were challenges that were encountered with the full field ERG, and the pattern ERG also does not do the thing that you want. Now, when you do a full field ERG, you are looking at the entire retina. If you have just bits of the retina that is in trouble, you can still get a good response because the rest of the retina is fine. Now, if you think of the visual field test, you are able to localize areas of discrete defects or lesions. So this idea was extended to the ERGs. And then they tried to do this stimulus. It is not exactly like what I show you here, right? It's more like this than that, but I couldn't get this one to animate. All right, so I'm just using this, but you will see this. And you can see that these ones have this hexagonal shape instead of a square shape, right? Basically, what it does is that each spot behaves like a small flash that comes on and goes off, all right? Now, again, they've done some wild mathematics on this thing. And at each point, the computer knows which parts of the screen turned on. And there's a topographical map. Of course, the average topographical map is somewhere in the system. So if, go back. Now I'm going forward. Wait. All right. So if you think about it and we say this side, let's call this side A, this side B, and that side C. Ignore everything else. So when this side comes on, and assume every other part of the retina is off, whatever signal is produced, the computer registers it to this part. Then this side will go off and another part will come on. And the computer registers that signal to that part. And so this on and off keeps going on at different parts. And the computer basically registers the signal at all these different parts. And at the end of the test, it puts it all together into a map. And you get this map. So you get sort of like tiny, brief flash ERGs that correspond to specific areas on the retina. You would notice that the sizes are not the same. Can anybody guess why the sizes of the the hexagonal shapes are not the same? The clue is they get bigger as they go out. It has to do with sensitivity of the photoreceptors. You are on the road, but not quite there. It has to do with sensitivity, but you are, there's a much, convergence. So in the retina, at the macula, you have more cells packed in a smaller area, while at the periphery, you have fewer. So it is scaled. It is scaled to capture essentially roughly the same number of cells. So as you go further into the periphery, they have to make the area bigger to capture the same number of cells that would have been captured in the center. Okay? All right, so it is scaled. All right, and usually we measure a larger area as well, around 60 degrees to about 120, depending on which size of the screen you are using. So the macula is about 20 degrees. All right, so the pattern ERG usually just has anything between 15 degrees and 20 degrees, so that it is just measuring the macular function. All right, now VEPs. VEPs are in terms of the stimulus. You have a question? Yeah. All right. Yeah.
My question is for the multifocal ARG, and if the points, the stimulus have to reflect a particular point on the retina, does the eye during the examination have to stay still so that probably the point doesn't come on the other side of the retina or anything?
Yes. So there's usually a fixation cross, and we expect the person to look at the fixation cross. Sorry. So there's a fixation cross. There's a point we expect the person to look at. The same way that we do visual field tests. So we expect the person to look at that point while the test is going on. Yeah. So the person has to fixate. Yeah. 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 The screen is no. They are not point dots. They are these areas. They are how do I call them? Hexagonal shapes. Yeah. That's why I brought this. So if you think of this guy, all right, it's like this. It's like this, but it doesn't have these square shapes. It rather has these hexagonal shapes. And it will be on a computer screen and it will be flipping. Do I have this one flipping? Yeah, it will be doing something like that. Okay? Parts of it will come on, parts of it will go off. And it's quite rapid. Yeah, I've slowed this one down. Yeah.
So I'm curious, does it record fixation losses just like the...
Yeah, so we have a big camera that will be looking at you. Yeah, there are a number of things you can do. Some of the systems are designed to stop taking data when the person looks away. Some have a camera that is focused on your eyes, and the person doing the test can watch to see when you are not fixating. Of course, there are also practical reasons why some people may not be able to fixate. If somebody has a big central scotoma, they can't see that, and so there are other things we ask them to do instead. Yeah. Yes. Hold on. Sorry, you were saying?
Thank you. So you mentioned that the patterns, when presented to the patient, is very rapid. But I want to know, are there situations where you slow the pace of these patterns to the patient?
Okay, so again, these are all different protocols. So you can do a whole lot of weird things with electrophysiology, depending on exactly what you want to probe. We are sticking to what is standard, okay? Just because that is what will be common, but we can do all sorts of weird things. Here you are seeing, for example, here you are seeing square, a rectangular side. There are stimuli that are round, like the dartboard. Yes, and there are all sorts of things we can do, but that's a good question. We can vary the speed. We can, but it depends on what it is we want to probe. Yeah. May I proceed? Okay. All right, so the VEPs also make use of a checkerboard pattern, and this one, we tap the signals from the back of the head, the occiput, okay? I won't go into details, but when you go down there, please ask them. We have these electrodes. I didn't talk about electrodes, sorry. Let me skip that. They will talk about electrodes, but essentially we need electrodes to be able to pick up the signals. And in the ERG, we put the electrodes at the bottom of the eyelid, lower eyelid. We put it on the lower eyelid, and that is where we pick the signal from. The electrode is made up of a high conducting cable. It's silver impregnated thread, and then we connect it to the machine. But I'll leave that for the electrode team when you go down there. How am I doing for time, please? I need to finish this by 9:30. 9:20. Okay. All right, the lecture has ended. All right, so then we have the VEP. So the VEP, again, like I told you, when you see N, when you see P, don't worry. So there are two types of the VEP. We have the flash VEP and then the transient, the pattern VEP. All right. What we'll be doing down there is the pattern VEP. Okay. Why do I have these white things appearing on? Are they part of my slide? Okay. I have no idea why they are there. Okay. So again, we are interested in the how do you call it? In the pattern VEP, we are interested in this P100, right? Which is the positive peak at 100. This tells us about the conduction, so the optic nerve. So now we've moved out of the retina and we are in the optic nerve headed towards the brain. Okay, so that's what this tells us. The disease we mostly use pattern VEPs for is optic neuritis. All right. The nice thing about optic neuritis is that the patient cannot see, the doctor cannot see. So we need to find out whether there's a problem there. And in optic neuritis, this P100 is very delayed. It's delayed. Okay. Something occurred to me a moment ago, but it has passed. Okay, the other thing, it's good for checking visual acuity as well. So this is what we usually use to check visual acuity in people. So when the check sizes are large and you can see them, you get a big signal. When the check sizes get to the point where it becomes blurred, everything becomes flat. And so the computer just calculates at which checkpoint did the signal become flat. And then it extrapolates that into a visual acuity measure. All right. Clinical applications, there'll be more by Prof. Sven and Heiko, so I won't talk too much about it. But you have someone with cataract or the media is opaque. Let me talk about the case we received. So we had a case of a trauma. So the case was referred to us. He had trauma. They couldn't see through the eye. And there was a question whether the eye was worth saving. Okay, so if the retina was still there and working, then they might want to take out the whole, take out the vitreous, take out the lens, and then probably fill it and put an IOL in there. So that might be a reason why you might want to do this, right? So the good thing about an ERG in this case is that, you know, the opacity makes a nice diffuser. So when you flash light inside, it scatters it all into the retina. So if the retina is alive, you'll get a signal. All right, so that is what we can use this for. Then what do I have here? Diabetic retinopathy, yes. So if you have, before you start seeing the aneurysms and stuff, you suspect, you know the person has diabetes and you want to know whether there's any problem going on in the inner retina, you can run your flash ERGs and multifocal ERGs, and then you would notice if there are some problems there. Glaucoma, I won't spend too much time on that, but the important takeaway home, the take-home message is this: when we think of glaucoma, we often think of, you know, the retinal ganglion cells, they are alive, and then suddenly, boom, like how you get an accident or you fall from a tall building and then you are dead. That's not actually what happens. It's more like they get sick and then eventually they die. So some studies were done and they could see that in this sick phase, if you intervene, you can actually reverse the trend. Okay? Yeah, and that is where IOP management comes in very, very useful. Okay. Now the problem is detecting this bit here. Okay. This is the problem. The problem is detecting this bit here. So electrophysiology can tell when you are losing signal. All right. And so they did some tests and they could tell it about, 4 to 8 years ahead of when OCT and VFT will find it. All right. Other conditions: retinitis pigmentosa, albinism, congenital retinal disease, retinal toxicity, chloroquine for treating lupus, optic atrophy, Leber's. I won't go too much into that. Unexplained visual loss, like I've been saying. This fascinates me. So I talk about it quite a lot. When the ophthalmoscope was invented, okay, before the ophthalmoscope was invented, we couldn't look into the eye. So unless the person had an obvious cataract, where you could tell that there was something wrong, or the person had a refractive error, and you put in glasses and the person could see, we didn't know what was wrong. And I think all of them were classified. I've forgotten the name they used to classify all those diseases. I think occult something. Okay. Then the ophthalmoscope came, and for the first time, they could see into the retina, and they could see whatever was going on, diabetic retinopathy, all of that. Okay, so the tool gives us something to probe our own, like, initial diagnosis. I have often thought, how have we, in Ghana, have we ever diagnosed congenital stationary night blindness before? Kolebu. But do we think that there's no such thing as congenital stationary night blindness among us? How do we properly diagnose staggards? How would we do it? So we have to infer and base it on a whole lot of things, yeah. And so one of the exciting things is that now if we suspect it's a staggard, we can actually just put the person behind the ERG and then check and actually confirm the diagnosis. Imagine how we used to diagnose glaucoma before OCTs and VFTs came. We looked at the optic nerve head in ophthalmoscopy to see how big it was. And we were probably treating people who just had large optic caps, who had no glaucoma. And we were letting people who had glaucoma go because the cap was not big enough. Then we had OCTs and we had VFTs. So there are a number of cases that we've probably looked at. We've never been sure. I think we can bring them back and then put them on this. And so this is the kit that is in Freiburg. You need specialized equipment. I showed you this just to show how it has developed. This kit. Sven, how old is this kit now? 20 years plus, yeah. But now we have moved on to handheld versions. All right. The kit you'll be seeing downstairs is one of the newer models, and we went for a full kit, so it's not as small as this, but it's fancier than this. But this machine is what has done a lot of the basic work. Like I was telling you, the ISCEV standards and all of that, a lot of it was done on this machine, and I was privileged to work in that lab and also gain my experience. So thank you for your time, and I think I made the 9:30. Did I? Okay. All right, so this is off the record, so we can pause the recording now. The recording can continue. Any questions? Yeah. How much is the setup? Wow.
I'm doing if someone wants to do the test.
Oh, to do the test. Oh, then that's your question.
I like answering this question. You are very kind, you are very considerate, you are very generous. So we just let you pay for the consumables. So currently VEPs are 200 CDs and ERGs are 500 CDs. A bouquet of it is 700 CDs. Very generous.
Okay, so Doc, thank you very much. I was waiting for this time to ask this question. That's the main reason why I even joined this program. So I'm an intern at Dr. Agawa Hospital, and we are facing a bit of challenge over there. The OCT we have and the VFT, mostly the VFT mostly doesn't match the OCT findings we have been getting. And it looks like all the time when you do the VFT, it comes out more positive, even when the OCT is okay. So even the new doctor there is no more working, even requesting for VFTs. So I don't know if there's anything this lab is trying to come out with.
Okay, so when you say the VFT is positive, you mean that the VFT is more, you mean the VFT tends to be normal? And the VFT is picking up more cases, and the OCT says, all right, so I would say I'm not too surprised. So we have our own OCT, and I've actually contacted Zeiss, and I've told them that they should give us access to their normative data. They have refused, but we will get there. We'll hack the machine. Don't go and tell them. Yeah. Like I said, we know that the normative database is not indigenous. It's not indigenous, and we suspect that that is the reason. But also check the setting. You can change the setting on the OCT to African. I'm not too sure you can do that on the VFT though, but that is part of the challenge. So you might want to check. But that's the thing. We are trying to build our own normative data for that. Yeah.
Doc, so from your own experience, if someone is referring to take an ERG and then maybe, okay, so let me use my experience. So there was a time I called my colleague, Dr. Adato, and then Dr. Delis about a case we saw at our place where the person was color deficient, and then I wanted to refer. So from your own experience, if I'm making the referral, would you advise that I specify that I need the pattern ERG or a full ERG? Like what specification should be made with certain conditions?
All right. So ideally, that is what we would have liked, that you have your tentative diagnosis, you've kind of done your differentials, and then you're asking us to do a particular test. But I think that would be too much to ask, because I think even in a place like University of Freiburg, where they've had the machine, as Sven will always say, they send down a patient, and you can tell from the notes that the doctor has no idea what he wants. He just wants you to do a test. So I think that's also why it's important that we at the lab also know how it works. And then when you give us, so we just say, give us the patient's history. Take a good history. It helps us cut down, like try to get the test so that at least we will start with the most probable test that is ideal. And if that doesn't work, then we work the other way. So try and give us the history as much as you can, and then we can try and work it out from there. Yeah. There's a question, Delis.
So please, in a condition that maybe you want to request for ERG,
yeah,
is there a need to do OCT, or ERG is more advanced, so you concentrate on just EIG?
Okay, so this is so ERGs are considered, we consider them as complementary tests. Okay, when you have, I think as a practitioner, most of your cases you'll be able to deal with them with OCT and VFT. And even just from the history, I think most of the cases, I'm saying that as a practitioner, most of the cases that you deal with, you're able to tell from the history, and by the time you've done OCT and VFT, you should probably know what it is that you are looking out for. Roughly, the cases that require ERGs are not, I would say, they won't make up the bulk of your practice. There will be relatively few. There will be your problematic cases, those ones that don't quite fit what it is that you are doing. The genetic cases, these we call, they call them occult maculopathies. Occult simply meaning that they are hidden. You know the person has a problem, but you just can't figure out what the problem is. That's what. So those will be the kind of cases. And usually by that time, you would have done your OCT and you'd have done your VFT. So I'll still encourage you, please still do all those regular things. Request your labs and do all those things. When none of them is giving you an indication, then we can come in. Yeah.
Thank you very much. So last month, we diagnosed a patient of retinitis pigmentosa. So I would like to know if the test can predict the number of years the person can go blind if there's no intervention. And also another question, can the test also give treatment guidelines that we should give the patient, because 13-year-old diagnosed with this and you can't do anything about it, it's kind of, yes.
The person I wish would be here to answer the question is going to be talking to you on video. She couldn't make a live, she had to be somewhere. She would have loved to take this. So one of our advisors, Professor Jaslyn Jolie, that's her job, really. And she's also part of ISCEV. I am not sure I know the answer to whether it can predict how long it will take for the person to go to blindness. Is that what you were saying? Yeah. But we did have a case of retinitis pigmentosa that was quite interesting. It took us a while to find it. Yeah. And the signal said there was a problem, but the retina wasn't telling us anything. So we had to take some retinal pictures of the extreme periphery, and then we found the bone speckle, the pigmentation. Yeah. So yes, so in that case, electrophysiology was handy. So we did see that there was a bad signal, but nothing on the retina in obvious view could tell us why we were seeing the signal we were seeing. But because we knew there was a problem, we dilated the person's eye, put them back on the retina camera, and went scanning all over the place, and eventually we did find it. There was another question you asked about treatment guidelines. Those would fall in the realms of back to you, the practitioners. The test doesn't say what you should do to the person. I mean, those treatment guidelines have long been established. Once you know what the condition is, the treatment guidelines have been established. But something interesting, because you have asked this question, is the issue about whether the drug is working. Okay. So if the drug is working, we expect one of two things. Best case scenario, you get malaria, you take chloroquine, even though your body might itch. When we check your plasmodium load, it goes down, and then you are back and you are healthy. That's our best case scenario. That is, we give you the medicine, we check again, the signals are back up, and you are functional. The situation we can also be satisfied with, it was going down, we intervened, and it has stayed like that. It also means the drug is working. Yeah. So ERGs, electrophysiology, is becoming more and more used in proving that drugs are working. You know, now that we are moving into all this personalized drug with gene therapy and all these things coming in, the question is it actually doing what it's supposed to do? And this is where electrophysiology is really, really strong, because it can tell, is function improving, or is function remaining the same, or is function declining? Yeah.
So my question is, I think the electrophysiology is more geared towards, I don't know if I think it's more for the practitioner benefits than the patient. Because I had 2 children from the same mother, and then we queried Stargardt, and we had them take a series of tests, the OCTs, the VFTs, the fundus photos and everything. And then we know, and from the symptoms, we know the person had Stargardt, but we still wanted them to come here for the test. And the mother asked us that if they come, will the children see better? And it's like, we know that even if we confirm the diagnosis, there's not much we can do. So because of that, we are thinking, so is it worth it for her to travel all the way to take this test? How do we convince patients to go ahead?
Yeah. This is not a scientific answer. This is my feelings and my thoughts. I am sober when I answer this question. The impossible, the difficult, we will do today. Now we have a machine, we can prove that the person has Stargardt. The impossible will take us a little bit longer, but even that one, we will do it. I went for a conference, and they have 14 PhD students. Do you know what they are doing? We have already solved the question as to whether we can grow retinas. We can grow retina. The problem now is getting the axons to go where they are supposed to go. And they have 14 PhDs working on that. So, yes, they have Stargardts. We might not be able to do anything about it now. but I have 28 people sitting here, highly intelligent, highly motivated. I think if they put their mind to it, we'll figure it out. So it's not going to happen today, but we can start thinking about it. Yeah, okay, so thank you very much. We've changed things around a little bit, so we are going to split straight away so that we can have more time on the machines. How many people do we have here now? I know a few people have joined from outside, so.